Good evening, everybody. Welcome back to daily lectures at New Age Cosmetic White EdTech Health and Technology. We are on the ninth chapter, which is a part of the second aspect of molecular biotechnology of microbial systems. So the ninth chapter would be protein therapeutics, and we move on to that now. With the advent of recombinant DNA technology, many of the properties of microorganisms that might be useful in a variety of applications can be more readily exploited. And in part 2, we examined some of the uses for genetically engineered microbial systems. Currently, bacteria are being genetically manipulated to act as biological factories for the production of pharmaceutical proteins, nucleic acids, and therapeutic agents, restriction endonucleases, chemical compounds, amino acids, antibiotics, and biopolymers. In some applications, cloned genes have been introduced into bacterial host cells to create novel biosynthetic pathways that produce novel metabolites. Genes and DNA fragments from pathogenic organisms have been isolated and used as probes for the diagnosis of disease in both animals and humans. In other instances, isolated genes and DNA fragments have been used to produce safer and more efficacious vaccines. Genetic manipulation of microbial systems also entails enhancing the natural ability of certain bacterial strains to carry out specific biological processes. For example, researchers have developed bacterial strains that can degrade environmental pollutants, improve the growth of plant crops, degrade cellulose biomass into utilizable low molecular weight compounds, and prevent the proliferation of specific insect pests. It is often assumed that the growth of large quantities of microbes is a routine procedure. Successful large-scale procedure of proteins synthesized by recombinant organisms, however, requires that many different factors be controlled during both the growth phase of the microorganisms and the purification process to ensure that high yields of a pure product are. And now we're on to the ninth chapter of molecular diagnostics. The success of modern medicine and agriculture often depends on the ability of workers in the field to detect the presence of specific viruses, bacteria, fungi, parasites, proteins, and small molecules in humans, animals, plants, water, and soil. Right? For example, the prevention, control, or treatment of infectious diseases is generally facilitated by the early and accurate identification of the causative pathogenic organism. Many of these detection procedures require the growth and culture of the potential pathogen and then the analysis of a spectrum of physiological properties that facilitate its identification. Although tests of this type are effective and reasonably specific, they are often slow and costly. These constraints apply to the identification of both bacterial and parasitic organisms. In addition, if the pathogenic organism does not grow well or cannot be cultivated, the opportunity to detect the disease-causing organism is severely limited. For example, Chlamydia trachomatis, an obligately intracellular bacterium, causes a sexually transmitted disease prevalent in North America and Europe. Clinical diagnosis of chlamydial infection is difficult because long-term cell culture is required, frequently false negative results, as the diagnosis of the absence of the organism is erroneous, are obtained, and consequently adequate treatment procedures are not implemented. Certainly, if growth were required for detection, then at the best only a few of all known pathogens or organisms, pathogenic organisms, could ever be routinely identified. To overcome this major constraint, molecular diagnostic procedures using either immunological or DNA detection methodologies have been devised. In general, any useful detection strategy must be specific, sensitive, and simple. Specificity means that the assay must yield a positive response for only the target organism or molecule. Sensitivity means that the diagnostic test must identify very small amounts of the target organism or molecule, even in the presence of potentially interfering organisms or substances. Simplicity is required for the tests to be run efficiently, effectively, and inexpensively on a routine basis. This is a comparison of the same methods to diagnose parasitic infection. A method would be microscopic examination, and then you have in vitro culture and mouse inoculation, detection of antibodies in serum, DNA hybridization PCR. So the first one, the advantages would be simple, direct detection of parasite, differentiates morphologically distinct organisms. Disadvantages, slow, laborious, and tedious, low sensitivity, cannot discriminate between similar organisms, requires a high skill level. In vitro culture and mouse inoculation detects only viable parasites, measures virulence and infectivity. The outcomes or disadvantages, slow and expensive. Different strains show a range of responses. Parasites may lose its viability in the specimen used as animals. Detection of antibody in serum, simple and fast, automatable, can be used to screen a large number of samples, not always specific, does not distinguish between active and latent infections. And DNA hybridization and PCR. Fast, sensitive, and specific, detects parasitic directly, can distinguish between species independent of various infections. Parasites need not be viable, it's automatable. Expensive, multi-step does not distinguish between live and dead organisms, possible false positives and false negatives. Continuing, in general, any useful detection strategy must be specific, sensitive, and simple. Specificity means that the assay must yield a positive response or only the target organism or molecule. Sensitivity means that the diagnostic test must identify very small amounts of the target organism or molecule, or even in the presence of other potentially interfering organisms or substances. Simplicity is required for the test to be run efficiently, effectively, and inexpensively on a routine basis. Some of the methods used to diagnose a comparison. This table shows a comparison of some of the methods used to diagnose parasite infections. Method, advantages and disadvantages. Microscopic examination. Advantages are simple, direct detection of parasite, differentiates morphologically distinct organisms. Disadvantages: slow, laborious, and tedious, low sensitivity, cannot discriminate between similar organisms, requires a high skill level. In vitro culture and mouse inoculation detects only viable parasites, measures virulence and effectivity. Slow and expensive, different strains show a range of responses. Parasite may lose its viability in the specimen uses animals. Detection of antibodies in serum, simple and fast, automatable, can be used to screen a large number of samples. Not always specific, does not always distinguish between active and latent infections. DNA hybridization and PCR. Fast, sensitive, and specific. Detects parasite directly, can distinguish different species independent of previous infections, and parasites need not be viable, automatable. Expensive and multi-step, does not distinguish between live and dead organisms, possible false positive and false negatives. This has been adapted from WEE's clinical and microbial articles and journals, courtesy to them. It is estimated that worldwide sales of innovative diagnostic accounted for about 77.7 billion in 1999. This figures continue to increase by 5 to 10% per year. The market for DNA-based diagnostic procedures was around $500 million in 1999 and is increasing at around 20 to 30% per year, so that in 2004 it was worth approximately $2 billion. In this chapter, the principles behind some of these molecular diagnostic procedures and the use of these procedures for a variety of applications are discussed. Immunological diagnostic procedures. Many immunological detection methods are sensitive, specific, and simple. They can be used for a wide range of applications. They can be used for a wide range of applications, including drug testing, assessment and monitoring of various characters, cancers, detection of specific metabolites, pathogen identification, and monitoring infectious agents. However, there are limitations. For example, if the target is a protein, then the use of antibodies requires that the genes contributing to the presence of the target site be expressed and that the target site not be masked or blocked in any way that would prevent the binding of the antibody. In principle, traditional diagnostic procedures for infectious agents rely on either a discrete set of traits characteristic of the pathogenic agent, or preferably one unique, readily distinguishable feature. The clinical microbiologist searches for the smallest number of biological characteristics that can complete certainty reveal the presence and precise identity of a pathogenic agent. For example, some infectious agents produce distinctive biochemical molecules. The problem is how to determine when the identifying component is present in a biological sample. Often such a marker molecule may be identified directly in a specialized biochemical assay that is very specific for the marker molecule. The problem with the approach is that it can potentially lead to a proliferation of highly individualized detection systems for different pathogenic organisms. A standardized method of identifying any key marker molecule, regardless of its chemical nature, is preferred, because antibodies bind with a high specificity to discrete target sites. Antigens, assays based solely on identifying specific body antigen complexes have abolished the need to devise a unique identification procedure for each particular marker molecule. ELISA. There are a number of different ways to determine whether the antibody has bound to its target antigen. The enzyme-linked immunosorbent assay ELISA is one method. It is frequently used for diagnostic detection. The ELISA procedure may be either indirect or direct. A generalized indirect ELISA protocol has the following steps. Bind the sample being tested for the presence of a specific molecule or organism to a solid support, such as a plastic microtiter plate, which usually contains 96 sample wells. Wash the support to remove unbound molecules. 2. Add a marker-specific antibody, primary antibody directed against the target antigen to the bound material, and wash the support to remove any unbound primary antibody. Add a second antibody or secondary antibody that binds specifically to the primary antibody and not to the target molecule. Bound conjugated to the secondary antibody is an enzyme such as alkaline phosphatase, peroxidase, or urease that can catalyze a reaction and converts a colorless substrate into a colored product. Wash the mixture to remove any unbound secondary antibody enzyme. Conjugate. 4. Add the colorless substrate. 5. Observe and measure the amount of colored product. If the primary antibody does not bind to a target site in the sample, the second washing step removes it. Consequently, the secondary antibody enzyme conjugate has nothing to bind to and is removed during the third washing step, and the final mixture remains colorless. Conversely, if the target site is present in the sample, then the primary antibody binds to it. The secondary antibody binds to the primary antibody, and the attached enzyme catalyzes the reaction to form an easily detected colored product. Since secondary antibodies that are complexed with an enzyme are available commercially, each new diagnostic test requires only a unique primary antibody. In addition, several secondary antibody molecules, each with several enzyme molecules attached, bind to one primary antibody molecule, thereby amplifying the intensity of the signal. With the direct ELISA protocol, figure 9.1b, a monoclonal antibody specific for the target antigen is first bound to the surface of the macrotire plate. This is an illustration of the secondary antibody, primary antibody, colorless substrate and colorless product, and macrotire plate. Macrotire plate, figure 9.1. Generalized ELISA protocol for detecting a target antigen. The primary antibody is obtained from rabbits that have been immunized with the target antigen, while the secondary antibody is from goats immunized with rabbit antibodies. The enzyme E is conjugated to the secondary antibody, A, indirect ELISA, or B, direct ELISA. And now to assess the amount of a particular antigen in a sample, the sample is added to the well of the macrotire plate and allowed to interact with the bound antibody. This is followed by a wash to remove any unbound molecules. Then the primary antibody and the secondary antibody conjugated to an enzyme are added, as described above, before the presence of bound antigen is visualized. The principal feature of the ELISA system is a specific binding of the primary antibody to the target site. If the target molecule is, for example, a protein, then a purified preparation is generally used to generate the antibodies that will be used to detect the target. The resulting antibody mixture, which is bound in the antiserum of an inoculate animal, usually a rabbit, contains a number of different antibodies that would each bind to a different antigenic determinant epitope on the target molecule. Such a mixture of antibodies is called a polyclonal preparation. For some diagnostic assays, the use of polyclonal antibodies has two drawbacks. One, the amounts of the different antibodies within a polyclonal preparation may vary from one batch to the next. And two, polyclonal antibodies cannot be used to distinguish between two similar targets. For example, when the difference between the pathogenic form target and the non-pathogenic one non-target is a single determinant. However, these problems can be overcome. Because it is not possible to generate an antibody preparation that is directed against a single antigenic determinant, namely a monoclonal antibody. Also, despite these drawbacks, diagnostic assays employ polyclonal antibodies are widely used for a variety of purposes. In mammals, a complex set of cellular systems is evolved to protect the body from toxic substances and invasion by infection agents. As part of the defense response, cells of the lymphatic system can be induced to produce specific proteins, antibodies that bind to foreign substances, antigens, and with the help of other immune systems, including the complement system, neutralize the biological impact. In response to an immunological challenge, each antibody-producing cell synthesizes and secretes a single antibody that recognizes with high affinity a discrete region, epitope, or antigenic determinant of the immunizing antigen. Because an antigen generally has several different epitopes, normally cells of the immune system. Each produce a different antibody against one of the many epitopes of the antigen. Such a set of antibodies, all of which react to the same antigen, is designated a polyclonal antibody. Early in the 20th century, although the polyclonal nature of antibodies was not appreciated, it was realized that antibody specificity could be used to prevent infections. Later, antibodies were used as diagnostic agents to determine the presence of toxic substances in clinical samples. Unfortunately, the effectiveness of polyclonal antibody preparation varies from batch to batch because in some immunizations certain antigen determinants for a particular antigen are strong simulators of antibody-producing cells, whereas at other times the immune system responds more actively to different epitopes of the same antigen. Also, individual animals often respond differently to a particular antigen. This variation can affect the abilities of different preparations to neutralize antigens because different epitopes have different potencies stimulating abilities. And one batch of polyclonal antibody may have a lower level of antibody molecules directed against a major epitope and not be as effective as a previous antibody preparation. Consequently, a fundamental objective of the applied use of antibodies as diagnostic agents or as components of therapeutic agents was to discover how to create a cell line that could be grown in culture that will produce a single type of antibody molecule, monoclonal antibody, with a high specific affinity for a specific target antigen. Such a cell line would provide a consistent and continuous source of identical antibody molecules. Unfortunately, the beta lymphocytes B cells that synthesize antibodies do not reproduce in culture. However, it was envisioned that a hybrid cell type could be created to solve this problem. This hybrid would have the B cell genetic components for producing antibodies and the cell division functions of a compatible cell type to enable the cells to grow in culture. It was known that normal B cell lymphocytes sometimes become cancer cells, myelomas, that acquire the ability to grow in culture while retaining many of the attributes of B cells. These myeloma cells, especially those that did not produce antibody molecules, became candidates for fusion with antibody-producing B cells. In the mid-1970s, these ideas became a reality. Formation and selection of hybrid cells. The initial step leading to the production of a hybrid cell line that produces a single antibody entails inoculation of mice with an antigen. To immunize. Now the illustration shows the mouse that were immunized, isolate spleen cells, myeloma cells, HGPRD, non-fused spleen cells, non-fused myeloma cells, fused spleen cells. Please take a screenshot of these. Now the HAT procedure for selecting hybrid spleen myeloma hybridoma cells. Several inoculations over a period of a few weeks. The animals are tested, generally using an ELISA or similar system. To determine whether they have developed an immune response. If they have, they are killed and their spleens are removed, washed, rinsed, and gently agitated to release individual cells, some of which are antibody-producing B cells. The splenic cell suspension is mixed with the suspension of myeloma cells that are genetically defective for the enzyme hypoxanthine guanine phosphoribosyltransferase, HGPRD. The combined cell suspensions are mixed with 35% polyethylene glycol for a few minutes and then transferred to a growth medium containing hypoxanthine, aminopterin, and thymidine HAT medium. The polyethylene glycol treatment facilitates fusion between cells. Nevertheless, the fusion events are rare and random. There will be myeloma cells, spleen cells, myeloma spleen fusion cells, myeloma fusion cells, and spleen fusion cells in the mixture. The HAT medium, however, allows only the myeloma spleen fusion cells to grow because none of the other cell types can proliferate in this medium. Unfused spleen cells and spleen fusion cells cannot grow in any culture medium. The HGPRD myeloma and the myeloma fusion cells cannot use hypoxanthine as a precursor for the biosynthesis of the purines, guanine, and adenine, which are of course essential for nucleic acid synthesis. However, they have a second naturally occurring pathway via purine biosynthesis that utilizes the enzyme dihydrofolate reductase. The verminopterin is included in the medium because it inhibits dihydrofolate reductase activity, hence AGPRD myeloma and myeloma fusion cells are unable to synthesize purines in HAD medium, so they die. The spleen myeloma fusion cells survive in HAD medium because the spleen cell contributes a functional HER-PLD.